We are again. I gotta do some more studying here. Okay, so glandular epithelium produces lots of molecules like hormones, stuff like that. Can produce hormones. If there's no epithelium, it's probably a bad thing, because that means that you've got a cavity that's probably not supposed to be there if there's no lining. Exocrine, the exocrine system sends molecules through the apical surface, meaning the top, while the endocrine system sends things through its basal surface, which is the bottom. Epithelial cells like to be next to each other. They like to have buddies. Okay, and the exocrine epithelial glands, I guess, glandular epithelium. You have unicellular cells. They have no ducts. And an example of this is the goblet cell, called the goblet cell for, I mean, it looks like a goblet, I guess. They're found in the respiratory tract, the gastrointestinal tract, and with H&E staining, they appear to be clear, colorless. But if you stain them with periodic Schiff staining, SCHIFF, Schiff, then they're magenta. Now, if you've got a fuchsin stain, FUCHSIN, fuchsin, then that will react with aldehyde groups, which gives a different color. Next, there's multicellular exocrine cells, I guess. I don't know, I'm just going back over my notes. Again, this is more for me, just a way I review. I force myself to review. Maybe I can watch these videos later. Hi, Mr. Goobers. Anyway, multicellular. These are not common cells. Mucus. These are, I think these include mucus secreting cells. Basically what they do is they cover the entire epithelium or epithelial sheet with crypts, like crevices. Like in the stomach, there's lots of surface area for digestion. I can't show the pictures either, because again, that's against copyright. Most multicellular exocrine glands have ducts to carry secreted material to specific locations. Like the esophagus has mucus glands. The sweat glands have serous. Okay, I guess, hold on. The esophagus has mucus material, mucosal material. Sweat glands have serous material, which is like more watery, I guess. Pancreas, same thing, serous, more watery. The trachea has seromucous secretions, which is probably a mix of both. And then the submandibular gland has also a mix of both. Hi, Antonio. A secretory unit is a basic cluster of secretory cells in the gland. Hey, everybody. Doing all right, just trying to study. So mucus secretions, mucosal secretions have increased sugar content in them, polysaccharides to be exact, and they have a decreased amount of protein. And of course, mucus, the purpose of it is protection, particle capture, like capturing foreign material that could cause infection and such, and also lubrication. Serous secretions have the opposite, decreased polysaccharide content and increased protein content. These secretions are found in relation to enzymes and antibacterial proteins. And secretory units can produce both. Doesn't have to be just doesn't have, wait, what am I, it doesn't have, oh yeah, it doesn't have to be just mucus or just serous secretions. It can be seromucus, like I was talking about earlier with the trachea and the submandibular gland, which I think is saliva, secretes some saliva. Yeah, okay. In epithelial tissue, there is something called the stroma, which is the supporting tissue, or like the matrix of an organ. Think about the liver, okay? You've got the connective tissue. It's all holding it together. That's stroma. And then you've got parenchyma. That is the functional part of the organ, like ducts or secretory units. For example, bile in the liver is one of the components of the liver's parenchyma, because the bile has a function, which is to emulsify fat and move along fat digestion. Okay, and then there's the apical portion of the epithelial tissue. We have microvilli, which are finger-like projections, very, very, very small, like 0.1 micrometers tall. Wait, no, I think it's 0.1 micrometers wide or something, times 1 micrometer tall. And they're covered by a plasma membrane. They are the main structural component in, oh wait, sorry. The main structural component of these microvilli is actin microfilaments. And what they do is they increase surface area to increase digestion. So they're found, one example is in the brush border of the intestine. There, hello everybody. Um, and why? Why in the intestine, right? Well, the point of the intestines is to digest food, right? You've got all these nutrients flowing through. Well, if they're just flowing through a tube with no villi, microvilli, then they're not going to be absorbed as quick. And the whole goal is to absorb as much as quickly as possible. And so all those extra villi, what they would do is increase the digestive capability of the intestines, because there's more surface area, more areas for those particles to touch in the intestines. And every part in those intestines has digestive capability. I mean, they're all epithelial cells. All these villi are still epithelial cells, from my understanding. And so they absorb nutrients just like the rest of the epithelium. However, the brush border is very, very difficult to see in pictures. I mean, if you just take a normal picture, you're not going to see it, of course. You have to use electron microscopy. Electron microscopes. Yep. Microvilli. There's something called a terminal web. It's connected to the cytoskeleton. Okay, and then there's stereocilia, which I think are made of actin. They have very long microvilli. These are up to 120 microns. That's a very big difference from the 1 micron that we were seeing earlier with those other microvilli. These are found in the epididymis or in sensory cells of the inner ear. And they don't, well, at least in the ear, they don't grow back. Okay, and then cilia. There's 2 types of cilia, it looks like. Modal cilia and non-modal. Motile, like moving. So the motile cilia are about 0.2 microns thick and about 5 to 10 microns long. They are made of microtubules, and there's 9 pairs encircling a central pair. It's like, it's very, wow. Wish I could show the picture, but again, I can't. It's like, it's weird. It almost looks like the transection of it, the cross section, kind of looks like a bullet. But yeah, they go around, like the cross section goes around like that. The 9 go around. Hi, Anna Maria. And then there's a pair in the middle. And there's diin protein, I guess, is the motor of that. Now, modal cilia are different from microvilli, and they're easy to confuse. Microvilli and stereocilia. Microvilli and stereocilia are both made of actin, but cilia, not stereocilia, but cilia, are made of microtubules. Microvilli is 1 to 3 micrometers. Stereocilia is up to 120 micrometers. and cilia are 5 to 10 micrometers. Or micrometers, however you say that. Now microvilli and stereocilia both create something called a terminal web, whereas the cilia creates something called a basal body. EM, what is that? I don't know what that is. Brush border. I don't know, there's all this EM slash LM. I don't know what that means. Okay, well, what the modal cilia do is they move fluid in one direction. Only one direction. It's unidirectional, which means one directional. And it's like there's a bunch of them, an army of them, and they all move it in the same direction. Now, non-modal cilia are they're really important for kidney homeostasis. When they're not in the kidney, problems happen. They're sometimes referred to as primary cilia, these non-modal cilia are, and apparently they are thought to exist on most epithelial cells. Yeah, they're made of microtubules. They're mostly chemosensory. For example, the renal epithelial cells. So these are like epithelial cells in the kidney. And then there's specialized olfactory cells. Olfactory meaning having to do with the sense of smell. And then some are mechanosensory. Yep. And then, okay, development. If they develop incorrectly, there's something called situs inversus, which is where organs end up on the other side of the body, which is not good. I mean, it's just, I don't know if it's not good. I didn't write that down, but it doesn't sound good, and maybe it causes problems. I don't know. Or polycystic kidney disease, which is, it's a disease, so it's not good. Now, what do I have left? There's a lot left. Oh well. So to summarize, microvilli are made of actin. They increase the surface area for where they are, for whatever purpose they have there. For example, the brush border of the intestines, right? Because they're trying to increase the surface area to increase digestion. Cilia are made of tubulin. That's not what it was saying up here. That's weird. Okay. There's modal cilia, and then there's immodal cilia. When it's modal, there's usually many, and they're directional. 9 plus 2. There's 2 in that center, and then 9 around. When it's immodal, that means it's solitary. It's by itself. Immodal, solitary, modal, many. Motile. Immotile. Not modal, motile. That's just my accent. Immotile are solitary, and these are referred to as primary cilia. 9 plus 0. There's just that 9. There's no one in the center. There's not a pair in the center. These immotile cilia are chemosensory, like renal tubules, renal tubule epithelium, or olfactory, and then mechanosensory. Vesicles, will usually be seen where the ships are docking. That made much more sense when my professor was talking about it. I can't remember what that's referring to now. I think it's something to do with, like, receptors. Or it's when the microvilli are moving and they're about to collect a nutrient. There's like a vesicle that eats up that nutrient. And the microvilli are there. I don't know. The basement, okay, now we're moving on to the basement membrane. Excuse me. The function of the basement membrane is to act as a barrier for the epithelium, and it's a selective barrier. For example, the glomerulus in the kidney. Glomerulus. Then it acts as an anchor for epithelium to support the stroma. Then it controls growth and differentiation. Sorry about that. Okay. The basement membrane is an anchor for the epithelium to the supporting stroma. It controls growth and differentiation. The basement membrane does. All epithelia have a basement membrane. It gives that epithelium almost a sense of direction of which way's up, which way's down. And knowing that, the cells know where to go when healing is needed in the body. This basement membrane also controls growth and differentiation. I already said that. Give me a second here. Got a message. Okay. The basement membrane is a barrier. But the differentiation problem, think of a hair cell, okay? Like, this hair is different from our scalp. It's all, there's differentiation there. But the basement membrane is a barrier. For example, the gastrointestinal tract is technically outside of the body, but it has an epithelium to protect the cells. Think about it. Again, I think I said this like 2 days ago or something. It's a through hole. Like, if you eat food, it's going to go all the way through your body, and eventually you're going to poop it out. But yet we don't get sick. I mean, unless it's food poisoning, but that's different. None of the food we eat is going to somehow end up in that state in somewhere else in our body. Like, you're not going to just do an x-ray and see a hamburger in my lungs, because that's just not going to happen unless I get a hamburger surgically implanted into my lungs. It's ridiculous. It's not going to happen, because your digestive tract is a through hole, which means it's got one entrance and one exit. Okay. Basement membrane. I've got some pictures here that I unfortunately can't show. They're pictures of light microscopy, microscope slides, H&E stains. So normally with light microscopes you can see one layer. Well, okay, looking at a trachea, you can see one layer. You can sometimes see one layer in ureters and in the urinary bladder. But with a transmission electron microscope, which is a much higher detail microscope and just more powerful, you can see three layers. The lamina lucida, which is lucida meaning translucent, that's the translucent layer. The lamina densa, densa meaning dense, that's the dense layer. And then the lamina fibroreticularis, which is meaning a fibrillar layer. I don't know how to say that word. Basically meaning fibrous. Fibrillar. That's how it's spelled. Fibrillar. Weird. I don't know how to say it, but it basically just means fibrous, I think. So there's three layers to that to that basement membrane. Whoa. Okay. And then you've got cellular adhesions in the epithelial layer. Give me a second. Okay. Where was I? So these cellular adhesions are basal and later portions of cells. Latter, I think, is what that's supposed to say. So these are anchor cells like hemidesmosomes and focal adhesions. These adhesions are there to compartmentalize the apex or the lumen from the base. They contain tight junctions, which are also referred to as the zonula occludens. And they are basically like a dam. They keep, like a dam, what a dam does is it keeps water out so that you can harvest it basically consistently, and it also keeps the water from going down in this area down here, if that makes sense. The cellular adhesions also stabilize the layer. Adherens junctions are referred to as the zonula adherens, or the belt, and then the desmosomes are referred to as the macula adherens. And then these cellular adhesions provide communication between cells in the layer via gap junctions. So zonula adherens and desmosomes both contain cadherins, or maybe it's cadherins, I don't know, probably cadherins. And they both contain zonula adherens, which are made of actin filaments, and then desmosomes, which are made of intermediate filaments. I think I'm going to touch on these a little later as well, because those terms sound familiar. Gap junctions, yeah, they also contain gap junctions. Man, I really needed to study this. I have almost no idea what I'm looking at. There's things called connexons. I don't know what those are. And I'm pretty sure that's it. There's just a bunch of abbreviations here now. Okay. I won't waste y'all's time with the abbreviations. Now we're on to the next lecture that I had, which... Okay. So, okay, yeah, we were in this other professor's class, and she was showing us some histologies. She was showing one necropsy where the lung was black, and it was like, well, it just didn't look right. And lungs, well, and this was this was histology, so it was a microscopic slide, and that lung tissue was black, or it was pink, and it's in the microscope slide. When it's stained a certain way, it's supposed to be clear. Lungs are supposed to be clear, right? So what they do is they do a biopsy, which means they basically just cut something off and then they send it to the lab for testing. Okay, histology is the study of tissues. You basically just take a hunk of tissue and put it onto a glass slide, put it under the microscope. But most of the time you stain it first. The most common stain in histology is H&E stains, which are hemotoxylin and eosin stains. What hemotoxylin does is it stains nucleic acid dark blue to purple. Eosin stains most proteins and positively charged structures pink. So connective tissue will show up pink because it's made from protein. Then there's other stains too. There's gram stains, acid-fast stains, silver stains, etc. Etc. But anyway, there's many parts to histology. There's the integumentary system, which means skin. There's the musculoskeletal system, respiratory system, etc. Most systems and organs contain a combination of different tissues. An organ is made up of 2 or more tissues. Otherwise, it's not an organ, right? You've got cells, tissues. What are tissues? Tissues are masses of cells. Then you've got organs. What are organs? Organs are masses of tissues, right? It's organization. Get it? Organize it. Yeah, okay. Skin contains, well, this is an example. So this is why skin is an organ. It contains 2 kinds of tissues. Epithelial cells clumps of epithelial cells, right? And connective tissue. So epithelial tissue and connective tissue has two tissues, so therefore it's an organ. At least. It's got more than two, I think. But what tissues have, okay, this is adding on to what I said earlier, they have cells and they also have extracellular matrix, and they've got a common function. And there's four basic tissue types. There's epithelium, connective tissue, muscle, and nervous tissue. With epithelial tissues, there's two types. There's linings. These are those cuboidal epithelium, cuboidal epithelium, simple columnar epithelium, stratified squamous, etc. Right? We talked about those earlier. And then there's glandular. So there's epithelial lining, and then there's glandular epithelium, which secrete things. Sometimes hormones, sometimes other things. But properties of epithelial tissues are that the cells are morphologically similar to each other, and they're usually very close together. I mean, just like with any tissue, but even more so in epithelial tissue, because there's little to no extracellular matrix. They're avascular. So there's a bunch of less clutter, if that makes sense. Now, epithelial cells help protect, absorb, and secrete things. They protect what they are lining. So, for example, the stomach epithelium, the lumen is the space in the stomach, but you've got the lining of the stomach is protecting the walls of the stomach from corrosion. You know, that acid could burn a hole in it if it doesn't have the right protection. And then they absorb. So think about the lining of the intestine, right? And then again, we talked about microvilli, cilia, all that stuff. And then secretion, glandular epithelium secretes. And it's finally, epithelial tissues are very adept at repairing themselves. If there's damage, there's a good chance it'll get repaired. But that differs in certain cells. For example, cartilage, which we'll talk about later. Okay, so epidermis is stratified squamous epithelium, while dermis is connective tissue, for example. And there's blood vessels in the dermis, but not in the epidermis, because the epidermis is epithelium. However, these epithelial cells want to be close, very, very close. Okay, then there's connective tissue. Let me see here. Ooh. Wow. Long way to go here. Connective tissue. It's often abbreviated CT. It binds tissues. It connects tissues, right? Not really surprised there. And it also forms capsules around some organs. It anchors epithelium and provides blood supply, with one exception, which is the cartilage. Right? Cartilage doesn't have blood supply. These connective tissue cells are either similar or dissimilar. These connective tissue cells contain lots of extracellular matrix. They function in support and binding, of course, connecting, right? And they're highly vascular, again, with cartilage being the exception. And most have pretty good repair potential. Now, in cases like the dermis, there may be some scarring, which is not good, but it'll still get repaired. Okay, to compare, epithelial cells are morphologically similar to each other, while connective tissue cells are similar or dissimilar. The epithelium has no extracellular matrix, while the connective tissue has lots of extracellular matrix. Epithelial cells function in protection, excretion, etc., while connective tissue functions in support and binding. Epithelial cells are avascular, while connective tissue cells are highly vascular, except for cartilage. And then the epithelial cells have good to excellent repair potential, while connective tissue has pretty good repair potential, except for cartilage. If you break cartilage, it doesn't repair very well, doesn't heal very well. There's lots of types of connective tissue. There's fibrous connective tissues. We have dense, loose, and reticular. There's special connective tissues: elastic, adipose, cartilage, bone, blood. Blood is considered a connective tissue. Excuse me. Then there's, well, okay, talking about fibrous connective tissue. There's dense, fibrous connective tissue, which is flexible and it has a high tensile strength. Decreased amount of cells, but an increased amount of extracellular fibers. Not as many nuclei, but it's got lots of protein. And then there's a loose fibrous connective tissue. It's like packing material. It looks more cellular relative to fewer extracellular fibers. So the dermis, for example, is dense connective tissue. The uterine endometrium, for example, is loose fibrous connective tissue. Reticular fibrous connective tissue is like a supporting meshwork. It's technically a loose connective tissue, but you can't see the extracellular fibers on H&E staining. So again, dense provides supportive framework. Loose also provides support, but allows for more movement. And then reticular is found in lymphoid organs, bone marrow, liver, etc. Components of fibrous connective tissue are extracellular matrix and heterogeneous cell populations. So components of extracellular matrix include stroma. Stroma is made up of fibrous proteins, collagen and elastin. Collagen is about 30 to 40% of the body's protein. That's a lot, considering how many proteins there are. Collagen has a high tensile strength, doesn't stretch very easily. And there's multiple types of collagen depending on how it's composed. Then we have elastin, which is stretchable, and the amount of elastin present depends on what connective tissue we're talking about. For example, there's lots of it in arterial walls to allow them to stretch. There's 4 types of collagen. Type 1 is the most abundant, and it's found in lots of organs. It's found in skin, bone, tendons, sclera, fascia, and organ capsules, and more, etc. Type 2 is amorphous appearance of collagen, which is found in cartilage, intervertebral discs, and the vitreous of the eye. Then there's type 3, which can only be viewed with special stains. These are like reticular fibers, etc. Then there's type 4, which are sheet-like layers found in basement membranes, kidney glomeruli, ocular lens capsules. And this collagen is non-fibrillar. Fibrillar. I don't know if that's how you say that, but I think it is. It's not fibrous. Okay, and then there's ground substance. This is like the goo in between cells and fibrous components. It acts like a cushion, and it's gel-like. Amorphous means it doesn't really have a shape. It's composed of water, glycosaminoglycans, which are referred to as GAGs, which include hyaluronic acid, I think that's how you say that, and chondroitin sulfates. And the 2 things I just mentioned, hyaluronic acid and chondroitin, are both good for, they're either good for your collagen or they're good for skin. Okay, chondroitin is good for your joints, according to lots of doctors. Hyaluronic acid, I've heard people use it on their skin, and they've reported good results. Okay, and then there's heterogeneous cell populations. We have resident cells and transient cells. Resident cells live a long time. They can live weeks to years. These include fibroblasts, reticular cells, adipose cells, macrophages, and mast cells. And then transient cells come and go as they need to, or as the body needs them to. These are neutrophils, eosinophils, basophils, and monocytes. Okay. And then there is mesenchymal cells, which are the stem cells for lots of different cell types. All right, these are stem cells for fibroblasts, adipose, cartilage, bone, muscle, and more. Fibroblasts are very important in connective tissue because they make the stroma. Fibroblasts make the stroma. Fibroblasts are responsible for secreting the majority of stromal and ground substance elements. For example, collagen, elastin, and those glycosaminoglycans. And then there's reticular cells, which are specialized fibroblasts. They secrete type 3 collagen, aka reticular fibers. These are important in lymphoid organs and also in bone marrow. And if you've ever seen proud flesh in horses, I'm pretty sure that's what these are. It's like a protective layer, I think. Okay, and then we've got adipose cells, fat cells. They're a special kind of connective tissue. They appear clear on H&E stain, and in most organs they're found, especially in the subcutis, right under the skin. They function, well, their function is shock absorption, energy storage, and heat insulation. Okay, we are done with that. Okay. Now, moving on to the next one. Okay, a lot of this is unimportant, but I'm going to be talking about immunology now. So, smallpox was the first disease to be eradicated. Vaccines are still medical procedures, so there could be consequences like adverse reactions. What the immune system does is it protects people from infectious diseases and foreign things. It's basically a widespread, complex network of interacting cells, cell products, and tissue that protects the body from pathogens and other foreign substances, destroys infected and malignant cells, and removes dying cells and cellular debris. There's all sorts of things it does, the immune system. And some of these things are autoimmune regulation, homeostasis, cancer surveillance, microbiome regulation, tissue repair and healing, etc. And there's a lot of problems that can be caused if the immune system's not working right, like autoimmune diseases, chronic inflammation, hypersensitivities, allergic reactions. Basically, ever since there's been life, there's always been something that's tried to be a parasite. There's always something that's tried to glean from something else. Not necessarily talking about human society here, but that's an example, right? The body is able to detect these parasites or these abnormalities, and as it got better and better, it became more and more complex, and that's what adaptive immunity basically is. Over time, the body has adapted to all sorts of stimuli. Okay, so innate immunity. There's innate immunity, and then there's adaptive immunity. Innate immunity includes chemical, physical barriers like the acidity in the stomach, granulocytes, which are also called phagocytes. They contain complement, they contain antimicrobial peptides, and they contain natural killer cells. Anything that's in a microbe that's not in the host itself, like bacterial cell wall components, can get attacked by the immune system. And then there's things called PRRs, pattern recognition receptors. These are things like toll-like receptors, TLRs, nod-like receptors, NLRs, RIG1-like receptors, I think, complement receptors. These are all pattern recognition receptors. Okay, talking about adaptive immunity, this includes lymphocytes, B cells, T cells, and they basically need to be activated by an exposure before they can give protection to the body. It can take a long time for acquired immunity to develop. Like, you can get sick, it can take you a week, it can take you a long time to get better. A week, maybe even a day, maybe longer. Might be in the hospital, you know? It's because your body's making the things it needs to fight these things off. And the way the body detects these threats is very, it's very concise. Sometimes it's down to the peptide sequence. The best part is that the adaptive immune system can learn, adapt, and remember things pertaining to pathogens and other microbes and anything foreign that enters the body. You've got receptors. Wow, there's a lot here. Ooh. B cell receptor, T cell receptor, all sorts of stuff. They both have antigen binding sites. They have variable regions, constant regions, transmembrane regions. Just different ways they attack foreign material. Everything in the body can be recognized by B and T cells eventually, because they're able to recognize hundreds of thousands of antigens, which are basically foreign particles. And then they're able to generate an antibody, which is their response to that antigen. A way of getting rid of it, a way of protecting the body. There's plenty of parts of the innate immune system and the adaptive immune system. The adaptive immune system includes memory B cells, plasma cells, memory T cells, cytotoxic T cells, helper T cells. And then the innate immune system contains things like mast cells, neutrophils, eosinophils, basophils, macrophages, dendritic cells, and other things. B cells, they recognize the shape of an antigen. They can bind to antigens in their native state, whereas T cells recognize peptides. They need other cells to digest the protein and present it to them, like the MHC, which is the major histocompatibility complex. Okay, then there's epitopes. An epitope is the specific part of an antigen that is recognized by the immune system. In other words, it's antigenic determinant. These are things like BCRs, TCRs. BCRs can bind to proteins, carbs, nucleic acids, and lipid molecules, while TCRs only recognize epitopes on proteins. And there's a bunch of pictures here I can't show. lymphocyte antigen receptors. Pretty much every T and B lymphocyte will recognize a different epitope. This means that their TCRs and BCRs are going to be unique. Lymphocytes are derived from the bone marrow by lymphoid precursors, and T cell precursors go to the thymus as they're developing their TCRs. However, birds have something called a gallinaceous birds have something called the bursa of Fabricius. What are gallinaceous birds? These are things like turkeys, chickens, quail, partridges, pheasants, and peacocks. So what are they? I don't know. They're characterized by their strong legs, short necks, relatively large heads. Okay. They're adapted to a terrestrial lifestyle, which means living on the earth. Interesting. And the bursa of Fabricius was how B cells were discovered. That's what they're named after, B bursa cells. And a similar organ to the bursa may exist in some fish. Now, primary lymphoid organs, these are the bone marrow, the thymus, the bursa, maybe some more. And in these, gene recombination is random. Receptors that recognize self-antigens are going to be generated, and then deletion, inactivation, or suppression of self-reactive lymphocytes are usually sufficient to avoid reactivity against self-tissues. There is some immunological tolerance here. Only a small number of lymphocytes will react against a pathogen because the body is so large and there's many locations of invasion. So how does a lymphocyte get alerted to that antigen? Well, they're always moving, and there's hubs around the body, lymph nodes, the spleen, liver, and the lymphatics connect all the lymph nodes. Lymph nodes are everywhere in the body, pretty much. Antigens and lymphocytes migrate to and from. And so this gives an increased chance of the lymphocyte meeting an antigen. What it does after it meets that antigen is it stops moving, and then it proliferates. It clones itself and creates an army of lymphocyte clones, clears the infection, and then memory cells are left over, which makes you stronger and better able to respond later to that same infection. It's like the military raising an army. All right, and that is it for the 20th of August. Thank you for watching.